Hello everyone, welcome in this channel and in this video. I am Dr. B. Krishna Prasad before you going to talk about bile. Bile is physiological fluid that is secreted from liver. Its color is dark green, color is dark green or yellowish brown. Yellowish brown. Daily secretion is 500 to 1 liter, 500 milliliter to 1000 milliliter is daily secretion. It is alkaline and isotonic fluid. Now composition of bile. Composition is, we can divide into two parts. Composition, water, that is 97% is water and 97% is water and 3% is solid substances in which there is organic and inorganic. Inorganic, bile acid, bile salts, bile pigments, cholesterol, lecithin, etc. are present, and inorganic electrolytes, sodium, potassium, calcium, magnesium, and this bicarbonate, chloride, etc. are present. This is composition of bile. Now, secretion of bile in liver takes place in two steps. First step is formation of bile, that is rich in bile salt, bile acid, bile salt, and cholesterol, etc. And second step, it is in duct, that is carrying bile. This bile in ductal cells contributes to sodium, bicarbonate, and water. Water will come passively, and this is second step. First step is in hepatocyte, a second step is in ductal cells. Now bile salts are added, and this is primary bile. Now it is primary bile. What is another type of bile? That is secondary bile. Primary bile is secreted from liver in two steps. Primary bile. And secondary bile is when this primary bile goes into the gallbladder, then in gallbladder it is concentrated. Water and electrolytes are absorbed and only organic substances are there. It is 5 times concentrated, and when this bile comes into the common bile duct by a squeeze contraction of gallbladder, then it is known as secondary bile. Now coming to types of bile acids. Bile acids are steroid acids formed from cholesterol. These are two types: primary bile acids, and secondary bile acids. Many types of bile acids are formed in the liver. Most important is cholic acid and chenodeoxycholic acid. Cholic acid and chenodeoxycholic acid, and when cholic acid and chenodeoxycholic acid enters into the small intestine, gut flora will convert it into deoxycholic acid and lithocholic acid. This is secondary bile acids. This is secondary bile acid formed into the gut, a small intestine, due to gut flora. This is primary acid and this is secondary bile acids. Bile acids are absorbable in a small intestine. Therefore, the conjugation occurs in the liver. Conjugation occurs in the liver by with glycine, cholic acid, chenodeoxycholic, for example, it will conjugate with choline, glycine. It will conjugate with glycine and form, this will conjugate and this will form glycocholic acid, glycocholic acid. And when it combines with then taurine, then it will form taurocholic acid. Taurocholic acid. Likewise, this chenodeoxycholic acid combines with glycine, then it will form glycochenodeoxycholic acid, and when taurine, then it will form taurochenodeoxycholic acid. These secondary bile acids also conjugate with these things. Now, these acids in presence of alkaline media combines with sodium and potassium to form salts. Sodium salt and potassium salt. This bile salt. Daily formation of bile salt is 0.2 to 0.4 grams and 4 grams, and total circulatory bile salts are 4 to 6 grams only. Total circulatory are 4 to 6 grams. This is about bile acid. Why bile is conjugated and formed into acid? Bile acid are destroyed, maybe destroyed in a small intestine, but these bile salts are resistant to action in a small intestine. It will go to the terminal ileum. It will go into the terminal ileum where it is absorbed. And after absorption, 90 to 95% will go into the intrahepatic circulation. Intrahepatic circulation. And from where it will circulate. Only 10 to 5% are absorbed in general circulation, out of which lithocholic acid is almost not absorbable and it will excreted in stool. But deoxycholic acid is absorbed and it will enter into general circulation and filter and in kidney and excreted in urine. It will go into the urine. So this is about bile salts, bile acids, bile salts and its circulation. Now this also contains one thing, bile pigment. Bile also contains bile pigment. Bile pigments are degraded product of hemoglobin. 1 gram of hemoglobin will form 40 milligram of bile pigment. There are two types, bilirubin and biliverdin. They are of no use in digestive function. So this is about bile. In next video, I will come with its functions and regulation of secretion and functions of bile and bile salt. Thank you very much. Please